am Dr. Danny Stroughton-Duncan, the Director of Education here at COLA. For those who are passionate about laboratory science and interested in staying up to date with the latest accreditation news and industry insights, Talk Lab to Me is an engaging discourse podcast hosted by COLA, a leading physician-directed accrediting organization that provides education and accreditation services to laboratories of all sizes. The podcast covers a wide range of topics, including the latest developments in laboratory science and medicine, as well as in-depth interviews with regulators and industry thought leaders about the importance of regulatory compliance. If you have any questions or comments about the podcast or the topics discussed, please feel free to contact us by filling out the form in the description. And don't forget to follow us and share. But before we get to that, are you looking to network with your colleagues? Do you need continuing education credits? Or do you just want to talk about your favorite technical topics? Join me at COLA's Laboratory Enrichment Forum, happening May 6th through the 8th, 2025 in Chandler, Arizona, where the focus will be laboratory science supporting the healthcare ecosystem. Welcome to Talk Lab to Me. Today we want to welcome my wonderful co-host, the Proficiency Testing Queen, COLA's own Maurisa Price. Welcome to Talk Lab to Me, Maurisa.
Thank you.
All right, but we have some guests here with us today. Tell us about our guests.
Sure. So today we have Lee Akerley. She's the Technical Director of the American Proficiency Institute, also known as API. As Technical Director, Lee is responsible for the technical operations of API's Proficiency Testing program. She has a bachelor's degree in medical laboratory science and has been with API for 12 years. Welcome, Lee.
Thank you.
You're welcome. We also have Sue Stiles with us. She is the Quality and Compliance Manager at API. She's responsible for API's compliance with CLIA and approved with CMS and other organizations. Sue has a background in statistics and international standards on proficiency testing and has been with API for over 20 years. Welcome, Sue.
Thank you. Glad to be here.
Okay, I got a question for everyone. All of you have been in proficiency testing for quite some time. So I saw Maurice, what do you love about proficiency testing?
I think PT serves a really important role in the laboratory in terms of identifying weaknesses and rooms for improvements and things that can really benefit the lab if they look at it from that standpoint. And I just enjoy being a part of that storyline. You know, you fail PT, I got you. However, what can we do to strengthen your systems so that whatever happened in PT isn't also trickling over with your patients and therefore improving patient care on the long run? So,
you know, we're here to talk lab to me. So we're going to get right into it. This is to our guest. We're all preparing for the CLIA PT revisions, which will be enforced in 2025. For laboratories, it's basically a matter of making sure they enroll for their 2025 in a 5-sample module for all of the regulated analytes, especially the 30 newly regulated analytes. But it's also important that all laboratories thoroughly read the instructions from their PT providers, because as you know, there are some very important changes to microbiology PT modules. Can you share with us about the process that PT providers have gone through to prepare for these changes?
Sure, I can answer that question. We have actually been preparing for quite some time. Even when the proposed changes came out before they were finalized, we took a look at our PT offerings. Like you mentioned, we have a lot of newly regulated analytes. So at that time, we ensured that we had 5-sample programs available for those analytes. So that was back in 2020. We also added a few more 5-sample programs to our 2024 offerings. So that left kind of some of the 2025 enrollment a little bit easier for us to get labs onto those 5 sample modules. And for 2025, we've actually looked at which programs we could discontinue 2 sample offerings. So this would be the majority of labs that may be participating in a regulated analyte. We did look at which programs we could eliminate. This mainly was in our chemistry discipline, where we had immunoassay, where a lot of those analytes will be newly regulated. So we discontinued the 2 sample program going into 2025. As you mentioned, microbiology, there are some changes. So for that, we do realize it's a unique discipline, especially in how it's scoring and the reporting of microbiology. But we did have to increase some of our bacteriology packages to include the new 2 susceptibility challenges. Bacteriology, the subspecialty under microbiology, labs usually have enough challenges. So that wasn't really a concern, or we didn't have to make many modifications to our bacteriology programs. But for our mycology and parasitology offerings, we did have to increase some of those to make sure that labs are compliant, since there's not always a lot of programs that we offer in those subspecialties. And we wanted to make sure that labs weren't going to have any issues. So that kind of sums up the program changes, but now we're currently working through internal systems and getting those grading criteria changes. So we have to kind of make a clean break from 2024 to 2025. So we have to continue with the grading criteria as it is for the remainder of 2024. Once 2025 comes, we will have all the new grading criteria for the new analytes and the currently regulated analytes, where the grading criteria will be changing, and we'll implement those as we roll into 2025.
Thank you for that. And like one thing we mentioned earlier is like when you're working with your proficiency testing, first thing is you want to make sure you're reading all your directions and everything like that. And it's already a little anxiety for some people on the bench to make sure they get that PT correct. But with all these changes, do you foresee any problems or difficulties for laboratories as they prepare to enroll in proficiency testing for 2025, just considering all the changes that are going on?
We're not anticipating any changes for the most part. We did look at our participants' enrollment. So we looked at what programs they're enrolled in, what analytes they're participating in, and we made some changes to the customers' renewals. So if they were, for instance, in a 2 sample program or immunoassay special reporting PTH, we had moved them over to the 5 sample program instead to help them make sure that they're compliant. But we also noted that for the customer. So if they look at their renewal, they will see that we had changed them to that 5 sample program. Most of the work we did take care of on our end, but we also want customers to look at their renewals thoroughly for 2025 to make sure that they're capturing all of the newly regulated analytes. Maybe they added any new tests to their test menu to make sure that their PT is reflective of their test menu going into 2025.
Excellent.
So from your perspective regarding enrollment, what are your expectations or guidance for laboratories, particularly when they failed to enroll in a PT or newly regulated analyte or the correct number of samples, or couldn't even get into the first testing cycle of 2025?
COLA has been working on our end since 2022 to get laboratories up to speed and informed of the expectations that we have for them. There has been 2 years of just constant education. Back in the third quarter of 2022, COLA released the COLA Insights, which is our newsletter, informing labs of the new final rule that came down and what the changes are going to be. Back in 23, for the first quarter, we sent out a technical bulletin, again, informing labs of the changes to PT regulations. back in September of 2024, we sent out another technical bulletin reminding the laboratories of the new PT rules. And then we also updated our primer with all the regulated analytes. In addition to all the education on our side, we've been doing a massive amount of testing, making sure our systems, our PT monitoring system is ready for that flip over of all the new regulated analytes that are coming on, plus the 5 that are going off, making sure that it can catch the pattern and spit out the right letters to our laboratories if they do have failures. So essentially, we've been working for 2 years trying to get this thing ready, and we're expecting our laboratories to have put in the equal amount of effort. So if we come into 2025 and they are not properly enrolled, or they do not have the correct number of samples, or they forgot to enroll altogether, they're going to receive a failure to participate score as they would any other time. When it comes to not able to obtain samples for the first event of 2025, that's going to be more of a case-by-case scenario, because if you are a laboratory that you've been testing, for example, testosterone, that's an analyte that's going from unregulated to regulated. You've been testing testosterone for 6 years, and then it comes almost time for that shipment to go out from API, and you're like, oh, we forgot to enroll in testosterone. I'm going to call API and say, hey, can you get me on that shipment that's going out next week? And API says, sorry, we don't have the samples to accommodate that. That laboratory, in my opinion, they're going to get the failure to participate. That delay in enrollment, that's on the laboratory. You just failed to enroll in a timely manner so that you can catch the first event. Whereas if it's a laboratory that they just finished validations and now the system is up and they're ready to start PT for a test that's new, and they call API to try to get enrolled, and API say we can accommodate you this first event, we'll set you up for the second and third event of this year. That laboratory, I would not hold them to the first event. They just started.
So what I'm hearing is that if you're a COLA accredited lab, you definitely need to go in your COLA Central, review those technical bulletins. And if you have done your validation and you are prepared for testing, you need to be contacting your proficiency testing provider, get enrolled, and ensure that enrollment. Is that correct?
The enrollment for 2025, from my understanding, and you can correct me if I'm wrong, Sue and Lee, it's now. Like, you guys can enroll in 2025 now. You do not have to wait till January to say, okay, let's enroll in 2025. You can go ahead and get yourself enrolled. Check your test menu, check what you're offering to your clients, check that against what you are enrolled to in in PT, and make sure you have an enrollment for every single regulated analyte. As for unregulated, if you participate in PT, make sure you have proper enrollment. If you're going to do splits, make sure you have that program set up. But yes, ma'am, this is not something you wait to the last minute to do. That should be done, in my opinion, the fourth quarter of the year for the next year.
All right, that's a little knowledge nugget for our audience out there. So if you haven't already, go through, check it, and make sure you are enrolled for your 2025.
So we've also had a lot of customer communication and resources available. As far as what you offer in your education to customers, do you have resources that help labs that might need guidance in how to achieve the successful performance for the newly graded or the new grading criteria itself, since some of those are already regulated? If they have kind of unexpected failures, what would labs expect to hear from you if they contact you?
Oh, absolutely. So as our COLA labs know, but if you're not familiar with COLA, any regulated analyte where you receive an unsatisfactory score, it goes out with a letter. We have quality improvement plans that go out with all our PT failures. API tells us you failed something, you're going to get a letter from us to say, hey, we're not aware that you failed this. And some of those letters are pretty much say, all the letters come with a checklist to help you try to identify the root cause and implement your corrective action so it's not a reoccurring issue. The first time around that happens for unsatisfactory, the first time failure, we pretty much just let you know that we know. However, you do not have to submit your responses back to COLA, because most labs really do resolve their first time failures without our intervention. If it's the second time initial unsuccessful PT performance where you have 2 failures for the same analyte, you'll get a similar letter say, okay, now this is the second time we had a failure for the specific analyte. Now we want to see your root cause and your corrective action. So that response, the laboratories do have to send back to us so that we can see what they're doing. Because at this point, we're trying to prevent a cease testing, because once you have repeated unsuccessful, unfortunately, it comes with a cease testing letter that is 6-month time out. We're really trying to catch the laboratories ahead of time before you make your way all the way to that third failure. Now, you guys can call COLA. We have Patty Street here. We also have Kim Irwin here. Those two people work behind me in the PT department when I'm not available. You can call us. If you call our number, you're going to get a person. If you fall into my voicemail, I promise you I will call you back. I have absolutely no problems getting on the phone with our laboratories and walking them through step by step on how to try to get them back to good performance if they have had failures, or just answering questions on how would COLA treat this. When you come into our laboratory, what would COLA do? Is this going to be a citation? I can walk you through all that, and I have no problem doing that. That is my job. So we do not mind the phone calls at all. If you have a question, call us. Again, you have COLA Central. There's a lot of resources there. We have a lot of primers there. We have a lot of templates there that you can utilize to help you with your PT issues there. But you got a live person, and I'm friendly. I'm not mean at all. Feel free to call COLA, and I'll be more than happy to assist you with any questions you have that's PT related that I can answer. All right, so here's a question for you. Prior to these upcoming changes, there had not been revisions to this list of regulated analytes for many, many years. So as CMS was preparing to propose the PT regulations revisions over these past several years, were the PT providers involved in that discussion? And if so, can you give me some examples of the kind of data and the guidance that you provided on CMS decision to add and delete analytes?
Yeah, I can handle this one because I was involved in the process, and it's a really interesting question to me because it was an opportunity where we actually were involved to a certain extent, and that's not always the case with these sorts of regulatory questions. So labs might be interested to know that CDC is really the science behind CLIA, and CMS is more the administrator. So I think CDC and CMS had more of the discussions, but it was really nice that they consulted PT providers as well. There's a CLIAC committee, the Clinical Laboratory Improvement Advisory Committee, that has physicians and researchers that advise CDC and CMS on all different aspects of CLIA. And most of the discussions took place through that committee with a lot of high-level expert involvement. And then they set up a working group around 2007 and had a list of recommendations that they sent in 2010 for how to move forward with choosing additional analytes, as well as reviewing the grading criteria and looking at changes in microbiology. They were really focused on all three of those areas. When the proposed changes were published in 2019, CMS outlined some of what that process was to add and delete analytes. For analytes, they looked at what tests PT was offered for, used PT provider catalogs and data that they had, and they cross-referenced that with Medicare data on test utilization rates to determine the most common test out there. And based on that, it was clear that a few analytes like primidone and NAPA could be removed from the regulations. So they didn't really ask PT providers a whole lot about that part. But they did survey us on how many participants we had for each of the new tests because they wouldn't have had access to that information. And they asked whether we had any comments on the list of tests that they were considering, whether certain analytes were gradable or not, and that sort of thing. Since the information on non-regulated analytes isn't reported to CMS or CDC, they didn't really have that much information about PT for those non-regulated analytes. So we were a source of information for them. But to me, the really interesting part was the grading criteria because they involved us a lot more in that part. And there's a lot more to think about with that part. They actually went through 3 rounds of asking PT providers to model different grading criteria. And what that means is that they gave us the grading criteria they were thinking of, and they asked us to go through our data and tell them what kinds of failure rates we would have if each of those grading criteria was used. So for example, if they're currently using 15% and they wanted to reduce it down to 12 or 10, they would ask us to rerun all of our data to the extent that we could and tell them how many failures we'd have. And it was clear that they were trying to find criteria that would not impact most labs, but would just set a consistent interval for results. So some of the old grading criteria, like if it was 30% of a mean, was clearly outdated from the 1980s. And the instruments are able to do a lot better now for some analytes. And so they wanted to find an interval that would reflect that current technology. They also wanted to know not just the failure rates for each sample, but also how many labs would fail 2 out of 3 samples, because that's where, as labs are in jeopardy and might have to cease testing if that happens twice in 3 events. And that could cause a lot of problems, even for the whole, not to be dramatic, but for the whole healthcare system if laboratories had to stop testing for certain analytes. So they didn't want to put something in regulation that was going to be a problem. They recognize that the research data, when you look at research, empirical research on acceptable error in patient testing and biological variation, might not correlate to the variation that happens with actual PT samples, where sometimes there's preservatives and things happen, or they're pooled from multiple donors. So they wanted to make sure that they use real PT data to see what would happen with what they had found through their research. They had to test that in real life. And then once the proposal was published in 2019, PT providers had the same opportunity as anyone else to comment on the proposed text. It was available online, announcements went out, and there were organizations and individual physicians and laboratories that responded. I think there were between 100 and 200 comments. And in 2022, when the regulations were finalized, CMS responded to a lot of those comments and even did make changes from the initial 2019 proposal. Like they proposed recommending viral susceptibility testing, and based on comments they got back, they realized that wasn't really very common and that they didn't need to do PT for that. So they said that they were removing that proposal from the initial, removing that analyte from the initial proposal. They also added some fixed amounts to some grading criteria where they made the percentage smaller, but it was going to be a little too tight at low ranges. So they added some fixed amounts to make sure that even at a low concentration, there would still be able to get a grade for participants, as opposed to not being able to grade or failing too many labs. And probably the most visible change that they made in response to feedback was they reduced the grading criteria for glycohemoglobin from the original 10% that they had proposed to 8%, because the diabetes community and endocrinologists, specialists in that area, said that 10% would be too much variability to allow in PT data. So I don't know that anybody ever 100% agrees with government work or decisions that are made in committees, but a lot of work went into the revision, and PT providers really did get to see a lot of that. And so we're cautiously optimistic that this will be an improvement. Hopefully there won't be any major drawbacks. We don't have anything that we're concerned about, or, biting our nails or anything. We really are cautiously optimistic that this will go well, and we're looking forward to it finally getting into place.
It's been a long journey. So.
Yes.
That's why I wanted to have those years to reference, because it has been a long journey.
Yeah.
Everybody knows that we've needed to update these regulations for a long time.
Yeah.
But the time was well spent. There was a lot of stuff that went into it.
I agree. So one thing I've always been curious about is how PT providers go about getting materials for PT challenges. Like, what's the process for selecting the materials for the challenges and establishing those acceptable ranges?
Yeah, I can take that question since I'm pretty closely involved in this process at API. So we don't actually manufacture our samples. We work with about 30 plus sample vendors that manufacture all of our samples for PT programs. These vendors, they should and most often do already manufacture other PT products or QC products. And we know that their practices are good. And each vendor usually supplies a specific discipline. So we don't have one vendor that's supplying a multitude of different disciplines. So these vendors are not only skilled in manufacturing, but they also are well versed in each of the specialties. So we have good technical experience on their end to help support the samples. And if we have any issues that arise, they can help support that product and make any changes or modifications that we might need to that product if needed. The materials themselves, we do have specific guidelines. We like to have it as close to a human specimen as possible. So we try to avoid anything that might need extra preparation where you might not with a patient, but sometimes that's not always unavoidable since these are manufactured samples. So we work with the vendors to select the target. So the PT provider is responsible for choosing what we will offer for the year. So we work ahead of time. So we've already done this work for 2025. Usually we wrap up about June where we select all the targets and then the vendors confirm that they can provide that material. So they will build that material to our specifications and they do testing before they release to make sure that it meets what we had asked for. As far as like the grading, how we determine any acceptable ranges for these materials. So even though we have specific targets, especially for quantitative, that's just a ballpark for us to have when we review the participant data. So all quantitative data, we use peer group consensus. So that's how the acceptable ranges are derived for quantitative analytes. Qualitative analytes, we do use that target for grading along with peer group consensus. So qualitatives, if we targeted it to be a positive sample, all peer groups should be detecting it as a positive. So then we use the peer group consensus to determine that that was the acceptable response.
That makes sense.
So with that being said about how we set the acceptable ranges, and we understand that laboratories need to be held to the new requirements, and you kind of said before that you can't offer much grace with, when it comes to meeting the new requirements because everybody's known that they are going into place. But do you anticipate that there'll be any changes in how successful performance might be viewed for the newly regulated analytes in early 2025? Will you have to wait for 2 failed test events with 5 samples before you issue a cease testing, or will you look at any previous PT records? Is there anything that labs might need to know or might wonder about that?
For the analytes that are unregulated as of right now, that will be enforced regulated in 2025. When I get my first file or the first event scores for 2025, this will be the very first time I have a score for that analyte. So if you fail it for the first event, the letter that will go out will be an unsatisfactory first time failure letter. I'm going to go based off of what the scores I received from API. So I don't have access to that historical data to see what you've been doing for the last, 2 to 3 years on, is this really a first failure or is it really a second failure? I don't have that access from my standpoint. So the first event of 2025 will be treated as the first event, and whatever score you get, you're going to be held to either a pass or a fail, and you'll be receiving an unsatisfactory letter. The only thing that I want to throw out there is our surveyors do have access to your historical data because they go in and they pull out all your books and they go back 2 years. So I can tell you that if our surveyors go in, and let's use testosterone as an example again, you've had 2 failures in 2024 already for testosterone, even though it was unregulated. And now you have a 3rd failure on the first event of 2025. Our surveyors are going to cease you because you still have met the requirements for cease testing. That will require that you just do your 2 consecutive passes and we can get you back reinstated. As long as you meet the reinstatement requirements, you'll come back out. So it's going to be, I don't know if I made that question really clear for our laboratories out there that's listening in. But again, here at headquarters, I don't have historical data. Whatever I receive from the PT providers, that is going to be what kicks out, whether that's the first time failure. Well, it should be a first time failure for the first event or passing. But when our surveyors go into the laboratory, they look back 2 years, and if you meet the requirements of cease testing, we will cease, and you will meet the reinstatement requirements in order to be reinstated. So, yeah.
Thank you. That's really helpful for us too, because we get a lot of questions from labs, and we're always telling them, call your inspector, call
Mm-hmm.
your accrediting organization, and they're always afraid to. And we
Mm-hmm.
tell them they'll be more of help to you than it will be of you asking us questions and us trying not to speculate, but yet trying to tell you what we may have heard.
Yeah.
We can't really state it as definitively or as clearly because we don't set those requirements. So
Exactly.
it's good to have you clarify that for labs.
Oh, absolutely. And trust me, they call and they tell me, well, they told me to call you all the time. Well, they told me to call you. I say, you're absolutely correct, because we're going to be the ones that come in, and we're going to be the ones that, unfortunately, cease or whatever we have to do. So I prefer the labs to call me and get it firsthand on how we're going to treat it. If we can catch it, get in front of it, and then go from there.
That's what we try to tell them, that it's always better to ask ahead of time. But I think they're always afraid that they don't want to offer themselves up.
Yeah.
They don't want to put themselves on the chopping block or reveal anything ahead of time. But the inspectors are going to find it anyway. So just
exactly
be open.
Just call us. We're nice.
Take the medicine now or take it later.
Oh, yeah.
And I think it's great, COLA taking the educational approach and not focusing on the fact of it being punitive. It's that at the end of the day, we're looking out for our patients, and that is the audience that we service. So we want to make sure that we are providing those accurate results, and this all comes in part with that. So, I like that idea of take the medicine now or later, anything to make sure that that patient is taken care of on the other side. Can you explain the process that you go through to decide if a PT challenge ends up being ungraded?
So you must mean when it was intended to be graded in the first place, right?
When it was, yes.
Okay.
Correct.
So that's one of the most common words even that you hear around our office. It gets a lot of attention. We're always thinking about ungraded because we're always trying to grade as many results as we can. CMS actually wants us to, CAP wants us to, and we feel like labs would rather be graded than ungraded because being ungraded creates more work. But it really is very situation specific. And the bottom line is that the data tells us when we can't grade. We need at least 10 labs to have reliable statistics, and CLIA says that we have to have at least 80% of the labs in the acceptable range based on the mean from that peer group. That's called consensus. And those two requirements come from CLIA, but we use them for every peer group, even for non-regulated analytes, because they prevent labs from being evaluated with poor data or questionable data that they might ask questions about. Why do you think that I didn't do a good job if I only got compared to 4 other labs? Well, that would be a valid question. So 10 labs and 80% of the labs in agreement. Poor agreement can happen for labs, things that are beyond the lab's control, like a reagent lot change that would make half the labs a little higher or lower than the other half. Or sometimes some instruments are less precise at high or low concentrations, so the data ends up being more spread out. And in those cases, we can end up not having that 80% consensus, and so we might end up not grading. So ungraded samples really fall into 3 categories. First, people might think if they're not graded, nobody was graded for that sample at all, but that's actually really rare. It would mean that we didn't get 80% of labs in the acceptable range for any of the peer groups or even for all the methods, and we'd have to investigate and document the reason for that for CMS and be able to advise labs on what they can do with the data that they got. So we try to make that not happen at all based on our history with each of the samples we're using and the way we work with the vendors. We have a lot of confidence in the samples, and it would really have to be some sort of sample problem in order for it to affect everybody, like some sort of contamination or some sort of unique stability problem that really doesn't happen very much. For qualitative analytes, it may happen a little bit more often because, for example, a microbiology challenge that is more difficult because we have to change around our targets every year, and there's certain categories of organisms that are required by CMS that can be a little bit harder. Labs may not see those organisms as often, but they have to be in the PT. So it can happen a little more often that we wouldn't get 80% of participants correctly identifying a microbiology target, but that's something that, again, we just go by the numbers. Rather than a whole sample being ungraded, what you guys might see at COLA more often is to just, and the labs would see this more often too, is to just have 1 or 2 peer groups that get ungraded because that group's results were spread out too much to meet that 80% consensus requirement. Sometimes, again, it's precision at a really low or high concentration that wouldn't affect other methods, but it does affect theirs. Maybe their method is recovering lower on the PT sample, and so they get into that area where the precision isn't as good, or they can always report that it's below their limit of detection, so that can happen as well. That doesn't usually affect consensus. But something like I mentioned earlier, an unrecognized reagent lot change, the manufacturers never expect much difference from lot to lot, but once in a while something will happen and the manufacturers will find out kind of if it happens right in the middle of a PT event, and the manufacturers probably start hearing from labs as well, because labs may notice that their patient values are differing also. If it happens right in the middle of a PT event, that can end up affecting that peer group, and then we can't grade. And kind of the third situation is just with an individual lab or a very small group of labs that don't get graded because they don't have 10 participants using their method. And if their method principle or their recovery or something is different from other methods and they can't be consistently compared to a larger peer group or to all the methods, then that small peer group might have to be ungraded. And sometimes that particular situation is one that goes on from test event to test event until there are more people using that reagent or that kit or something. So we know it creates more work for labs. We're always researching method similarities so we can provide peer group comparisons and not have those ungradeds happen quite as often. And as I mentioned earlier, CMS also watches ungraded samples really closely. We have to provide explanations and reports to CMS every year as part of our annual application process. So we really have to have solid documentation. It's not a subjective decision. And sometimes when labs call us, they think that we can just change our decision or that they can change our mind or convince us. And sometimes we do get helpful information from laboratories, but a lot of times it's just based on the data and there isn't much that we can do. So we try to grade whenever we can, but hopefully that clarifies or demystifies it a little bit.
It does. So I have another question similar in some of the natures about the grading, but can you just break down the process for a challenge that's graded under all methods group versus a specific peer group?
That's an interesting one too. I love all this data stuff. The default really is that almost all results are graded by peer group. Labs just prefer to see that their results agree with other people using the same method because it takes one variable out of the equation, right? If it just happens that their result didn't agree with their own peer group, they don't have to say, well, where did this data come from and what methods did it come from? But if a certain peer group doesn't have 10 participants or doesn't have that 80% consensus, then that peer group can't be graded with those statistics, even though other peer groups might be graded at their own peer group. But for that one that didn't get consensus, we have broader groups set up that will calculate statistics for a larger group using the similar methods instead of the exact same method. Like labs that all use the same reagent, even if they use different instruments, we would calculate stats that way and see if there's consensus, and then we might be able to grade those labs that didn't get consensus at their identical peer group with a little bit bigger peer group. So not everybody always gets graded at the same level. And if that larger group also doesn't reach consensus, ultimately the all methods group is the last resort. If that all methods group gets consensus, then the participants in that smaller peer group would get graded against that larger amount of data. But that doesn't mean that everybody got graded that way because we're trying to go with the peer group for as much as possible. The means are just more comparable with the results that people are getting in their own lab. So that's kind of a simplified explanation. It's easier to explain with a certain laboratory situation, and our technical support staff is definitely willing to walk through that with a laboratory to explain why there wasn't consensus or why they were graded with a particular peer group. And then there are some analytes, I don't know if you were thinking about this or not, but like blood bank or microbiology cultures where peer groups don't really apply. If it's a microbiology culture, everybody should be expected to get the same result. There's no reason to allow for any differences by method. So those aren't peer groups. Those are always all method grading.
Yeah, it's really good to know. And I feel like we just took a look behind the curtain. Like I've always wondered what went into, the background of pulling that together. So thank you for sharing that.
Yeah, it's really fascinating internally too. So it's kind of fun to think of how to describe that to someone.
Well, let me ask you this. Since we're on this topic, If a laboratory is shopping around, because sometimes when I see they have that exemption where they're not being really graded and it's like the artificial 100 for the whole year after year after year, my thing I'm usually telling laboratories is see if you can find your peer group. There may be a peer group somewhere that all your little family is aggregating to. Could laboratories call API, for example, and say, I'm using this instrument with this reagent. How many peers do you have in your database so they can know whether that's going to be the best PT provider for the analyte or whether they need to shop around?
Yeah, we can actually help with that. So if they call tech support, we can definitely walk them through some of that. Although we do a lot of investigations on our own as is. So if we do have smaller peer groups, we do contact manufacturers and have good relationships with them to see, does this method compare to this? Can we make a larger group? So we really don't like to have those smaller groups of labs go ungraded over and over again. But sometimes that's unavoidable, just depending on the method. Sometimes they're older methods that are no longer supported. So we're not always able to help the lab, but where we do, we actually are doing it up front. But a lab can certainly contact us and ask us, even if they're looking for a different instrument, we can help them with peer group data or show them where to find maybe which are the larger peer groups that might be more favorable for them to look at that instrumentation. So we're always willing to help and have that discussion, but it's not always something that we can work around.
Yeah. Okay. Good to know.
So this question has had a little buzz in my network about these WBC changes. So one of the most significant changes starting in 2025 is laboratories that perform and report both automated and manual white blood cell differentials will be required to enroll in proficiency testing for both. What guidance does API have to help laboratories understand the modules that they need to enroll in for this? And also, do you expect that laboratories will have quite a bit of questions about this change?
Well, our guidance is really pretty simple. We used to have two blood cell ID programs, one that had CMS scored in the name, and the other one had not CMS scored right in the name of the program. And laboratories would choose the CMS scored program if they wanted their blood cell ID to go to CMS, or the not CMS scored if they preferred that their automated differential went to CMS. It was actually harder to explain in the past than it's going to be now, because having the automated differential want to go to CMS was a pretty common choice. So they had more questions before about how were they supposed to enroll in their blood cell ID. So now that not CMS scored blood cell ID program, we discontinued that for 2025. And the current program, the only one that's available, says CMS scored on it. So we don't think that labs will have many questions about this. We automatically put everybody into that CMS scored blood cell ID program, and their automated differential scores will automatically go to CMS. So they really don't have any choices they have to make. It was more confusing before when they didn't realize that one or the other got sent to CMS. So this is a change that we feel like is a lot clearer for labs. The way CLIA is doing it now. It requires some changes in the background for all of us and changes in how surveyors are looking at results maybe, but the only question maybe that might come up is whether they have to really do blood cell ID testing or not. We did have a lab call the other day that they weren't sure if what they did with their slides required them to do PT. And so we would refer them back to their inspector, their surveyor, their accrediting body, whatever the situation is, because that's not a decision that we can make. But for most labs, the way they do their slides and the way they do their differentials, it should be pretty clear now that they're enrolling in PT for both.
I agree. I think this change was a good one. Many laboratories have been enrolled in PT for both the auto differential and the cell ID anyway. Starting in 2025, I'll be able to see both set of scores, and there will actually be less confusion about which procedure the scores represent.
Yeah, to your point, I wasn't really thinking about the evaluation side of things, but you're right that a lot of laboratories already thought that both of those scores were going to their accrediting agency. So we felt like it was our responsibility to tell them what the changes are. We didn't want to leave out any changes when we told them about changes, but I think it was confusing that they didn't really feel like it was a change. So
Like, what you mean? I do both anyway.
yeah, it was a
Yeah.
little bit hard to explain.
Yeah. All right. So let me ask you this. In terms of shipment of PT samples to very remote areas, when you do that shipment, what steps do you go through to ensure that sample integrity is maintained through the shipping process? And can you give me some examples?
Sure, I'll try. I've been having a lot of discussions around our group about stability. We've been working on stability documentation and talking about our packaging and everything.
Mhm.
And we don't turn any CLIA labs away. So we do ship to remote areas, even if someone's on an island off the coast of Alaska or a
Mhm.
US possession like Guam. Those labs also have to buy controls, and usually our samples are similar to controls in terms of stability. There are sometimes preservatives added, and the manufacturers have done stability studies to set an expiration date. And we really watch those expiration dates carefully. The manufacturers set them very conservatively, and they understand that the samples all have to give the same results all the way through the sample's lifetime. And they also understand that they're going to be shipped. So we work really closely with the vendors to have the freshest samples possible, and then we store them in colder storage at API than they might need to be during shipping or during storage at the lab. Sometimes we store them frozen. We store almost everything refrigerated and have electronic temperature monitoring the same way laboratories do, because we want to make sure that they get stored exactly the way they were supposed to be stored according to the manufacturer. So that helps prevent anything from happening before they go out and ensures that everything's in the best condition it could be. And then it's kind of a misconception that the samples need to stay cold all the way to the lab. In most cases, they're not fresh patient samples, so they are a little sturdier than the fresh patient samples. The ice pack is just to prevent extreme heat, and too much ice could freeze them. So when we're sending it somewhere remote like Alaska or Guam, it would be worse to have hematology samples freeze. We've actually seen what happens when that happens. So we have backed away from adding extra ice. The best way for us to really work with those labs is to understand how the samples get there. And we'll adjust our shipping process if we need to. Like for an island in Alaska, we'll find out what days of the week the plane goes to their island, and then we will set our shipping so that we send the kit out on the day it needs to get to the airport where the plane's going to leave, because we don't want to miss that flight, just like you don't want to miss the flight when you're traveling somewhere.
Yeah.
We don't want your samples to miss their flight to your island. So we work with labs that way. That really has been the most effective way to do it. Once in a while, we will use additional ice if it would help, but it's really quite rare. And most of the locations UPS or FedEx can get to in 3 to 4 days. We don't have to worry as much as labs think. If they feel that they had a really hard time, if they know that, the samples took longer than that, they can always call us. We have a replacement deadline when they can get free samples if they call and need help before that replacement deadline and clearly something has happened. Our customer service staff can get new samples out to them. Our technical support staff can talk about the samples, look at them visually, sometimes hematology or blood bank. You can see if they look okay. We could send a replacement if it was needed, but the labs could also indicate a comment if they decide that, yeah, we're going to go ahead and test these samples, and just enter a comment so that way, if their results do look like they're significantly different from the other results, we can look at that comment during the grading process as documentation. And then we do, we don't want labs to feel like they're just out there on the hook. We do look at our data distribution for any signs of instability to ensure that the samples are performing the way manufacturers expect. So if it's, the heat wave in Arizona where it's been 100 degrees for 100 days in a row, we trust our shipping and we trust the samples, but we are looking at the data so that if there was something going on that affects more than just one lab, we would be able to tell. So it's something that's always on our mind.
That's good to know. Okay, so that you kind of answered my next question. So just wanted to clarify, because when I get a laboratory sometimes, and they call me retroactively, like they'll tell me, I received these samples warm, they were really hot upon receipt, they sat on our shipping dock, shipping dock for a second, and all these reasons to why, and they had these really terrible scores. And then they said, well, and it's not necessarily API, but they said, I called my PT provider and they told me just put them in the refrigerator, let them cool down, and then I could test them. And so I'm sitting there listening to that, and I'm just like, well, if that was a patient sample, would you have just thrown it in the refrigerator, let it cool down, and then ran it? They're like, no, we would have rejected it. And so I usually say, okay, well, you're supposed to treat PT in the same manner as patients to an extent. So why didn't you just put in your exception code or give them, there should be places where you could put comments and things of that nature. So that's usually my advice when I'm talking to a laboratory. So hearing what you're just telling me, they can actually still run the sample, put in the comments. If they fail, do that failure still stands, or do you go back to the comment like, okay, well, they said it was received warm. Do they get that exemption score that I usually receive with the code 4 or what have you? What does that look like when they put in that comment versus actually putting in what I call code 11, where they're saying, I can't test this for whatever reason they pick? Or is it the same?
Yeah, so I can help answer that question. So when they put in a comment, which we often instruct to, so if a lab calls us and said, something happened with their sample, any exceptions, we do tell them to put in a comment. We do say they can, of course, run the sample as they would a patient, put in the comment. Our technical staff does review all of those comments. So most of the time, we just leave the comment in our system as is. So then when we go through the grading process, what happens is the comment will be applied if the lab may potentially get a failure. So we're not going to fail labs who may be outside the acceptable range if they've put in that comment, but we don't always scrutinize the comments and decide whether it was a valid complaint or not. We kind of leave that up to their accrediting agency to review when they come in for a survey to make sure that you felt that the lab was making the right call and that the outcome was what you expected. So that's how we handle them.
Okay.
And when it comes to those footnote codes that are on some of the samples, Marisa, you mentioned like a code 4 that comes through with the data. That's really what that means in CMS terms is an exception was requested.
Right.
So we always tell laboratories that they can, put a code or a comment on their sample, but it's just a request. We don't feel like we have the authority to say, oh yeah, you're absolutely okay, unless the data looked okay. So it's always up to inspectors, like with the hurricanes, that we are allowed to, we get permission from CMS sometimes to put a code on there for a natural disaster, but the surveyors are still going to review and find out that you were really only closed for one day. You were open the whole rest of the time. So there might be a code on that score, but it's always up to the accrediting body or the state inspector what ultimately should be done.
Understood. Yeah.
Sue and Lee, this was just so much wonderful information. Like I said, I feel like I now know the behind the scenes, what goes on from a proficiency testing provider side. So I want to thank you both for that information. As we close out, is there any final thoughts you want to leave us with?
I would mainly want to say thank you for inviting us. It's always fun to talk about PT, and I don't get a lot of opportunities to talk to people about PT who understand it and live it the way we do. But I also want to say that we really appreciate working with COLA. We like that you have a similar style to we do, that you interact with laboratories and that you're open and, take phone calls personally and all of that, because we feel like being friendly and open to discussion and willing to listen to a laboratory is the best way to have that educational approach that you talked about that really aligns well with API's view as well.
Thank you. And I want to give a big thank you to my co-host, Marisa. And she is, as she said, she's approachable. Our Cola Labs can reach out to us. She is here for your questions and to share all that wonderful proficiency testing knowledge. Do you want to leave us with any last thoughts?
This was an excellent conversation, and I hope we've been able to address some of the uncertainties the laboratories may have been struggling with. Our laboratories know that they can call me if they still have questions about the new proficiency testing regulations. I am here to help. I'd like to thank Lee and Sue for all the great information you shared with us today. And thank you, Danny, for inviting me to participate.
Oh, you are very welcome. And, thank you for talking lab to me. So until next time, stay compliant, stay curious, keep talking lab to me.